Hello, everyone. You're once again recorded. Welcome to BIO191 General Practical Biology 1 online class. Last time we examined how to procure plant and animal material for practical exercise in the lab. Today we go a step further by looking at how to prepare temporary slides. At this juncture, I'll start sharing my screen for better understanding. Our topic once again today is preparation of temporary slides. Expectations of the lesson is that's the expected outcome is that on completion of this lesson, you'll be able to prepare the epidermis of onion leaf, epidermal cells from named leaves, and mouth epithelial cells. General methods of making a temporary preparation as shown in this diagram. First, we place a cell suspension in a liquid on a slide. Then, if the material to be studied is dry, place it directly on the slide and adding water, glycerin, or stain to it. Then cover it with a cover slip. Be careful not to trap air bubbles while lowering the cover slip over the material, as displayed in the diagram here. The requirements, materials that will be provided for you are microscopes, magnifying lenses, lens tissue, specimen slides, cover slip, methylene blue, iodine or 1% acetic acid, water, onion bulb, leaf of a named plant, e.g., elodia or mango, toothpicks, sharp razor blade, and forceps. The first we'll examine is preparation of temporary slide of epidermis of a leaf. The procedure is, first and foremost, place a drop of water in the middle of the glass slide with a mounted needle or a pointed camel head brush. Transfer a small strip of onion skin to the water. Now, place a thin glass cover slip over the object. Examine the preparation under the low and hyper objective. Stain with 1 or 2 drops of iodine. Then you examine under both low and hyper objectives. Repeat the staining process with a fresh mount using grams iodine provided. Finally, stain a fresh mount with methylene blue. Examine the preparation under the low and hyper objectives. Draw the specimen under hyper objective after selecting the stain that reveals many of the internal components of the cell, as seen in the diagram 2, figure 2 displayed there. Those are some of the structures you will see. You will see the nuclei, you see cell walls, and the cytoplasm as a whole. First activity you are to attempt is to suggest some reason for the ultra-structure and transparent nature of epidermis cell. Can you observe any trace of chlorophyll in your specimen? Give reasons for your answer. Now, let's see the second preparation. Secondly, we are going to examine the epidermal cells from a named leaves. Procedure. First, place a drop of water on the center of a clean glass slide. With the forceps, remove a leaf epidermis from the plant and place it on the drop of water on the slide. Care must be taken to mount the upper epidermis with its upper side upwards and the lower epidermis with its lower side upwards. Carefully place the cover slip over the drop of water and the leaf. Then place the slide on the stage of the microscope with the leaves directly over the opening of the stage. Stain the slide as necessary to show some of the internal structures, examine with both the high and low power objectives, then draw a label under the high power objective. So these are what you will observe for both the high and for both the lower and upper epidermis. Notice that the lower epidermis is more visible. There we have many stomata, that's the opening, and guard cells surrounding them, through which water moves in and out of the plants. Now, list the differences between the cells of the upper and lower epidermis of the leaf, and what are the functions of the specialized cells observed on the lower surface of the leaf. Then state the differences in shape and arrangement between the onion cells and the cells of the leaf. Then the third and the final one we look at is the mouth epithelial cell. We observe onion, which is a plant, leaf, which is a plant. Now we are going to look at mouth cells, epithelial cells of the mouth of man, who is an animal. First, place a drop of water on the center of a clean glass slide using a broad, flat end of the toothpick or a blunt, clean scalpel. Gently scrape some of the epithelial cells from the inside of your mouth. Stir the water on the slide with the end of the toothpick or scalpel to mix the cheek cells with the water. Then dispose the toothpick or scalpel. So at this point in time, the smell should be made thin by adding a drop of isotonic saline solution, which is 0.9% NaCl to it, before putting the cover slip. Examine with low and high power objectives. The stem with 1 drop of methylene blue. Examine with both high and low power objective, then draw and label the cheek cells under the high power objective. So these are the kind of structures you will see there. In conclusion, you have learned how to prepare slides of samples that will be studied under the microscope. Learn to distinguish between plant and animal cells. However, the general reasons for making slides is to make the specimen more visible or render the details more clearly. Activities. See the activity displayed. What is the shape of the cheek cell? How are plant and animal cells similar in structure? How are plant and animal cells different in structure? Next class, we'll be looking at techniques for microbial structure and gram staining. Thank you very much for listening. At this junction, I'll stop sharing my screen. Thank you for participating in one way or the other. Like to let us know that the program, practical classes for BIO191 General Practical Biology 1 is already ongoing at the 13 designated center, North Central, Abuja and Makurdi, Northeast, Yola and Bauchi, Northwest, Kano and Sokoto, Southeast, Enugu and Owerri, South-South, Uyo and Benin, and finally Southwest, Lagos, Abeokuta, and Osogbo. Thank you very much for being part of this. Broadcast it to your classmates and friends around. Let them be part of the practical of a truth very, very important, because it will be used for your final assessment. Thank you very much. Bye, and see you next time.